After DNA isolation, we can analyze them or visualize them by using gel electrophoresis. And one of the type of gel electrophoresis, it is agarose gel electrophoresis. And this agarose, it is very convenient for isolating DNA fragments that are ranging from a few hundred base pairs to 20 kilo base pairs. So agarose को इस्तेमाल करते हुए हम छोटे से छोटे फ्रेगमेंट को भी आइसोलेट कर सकते हैं और ऐसे फ्रेगमेंट को भी जिसकी लेंथ जो है वो thousands of base pairs होती है. Now what is agarose? Agarose basically एक एक्सट्रैक्ट है सीवीड का या एल्गी जो कि मेरीन एनवायरनमेंट में या ओशंस में ग्रो करते हैं. उनसे इसको एक्सट्रैक्ट किया जाता है और पाउडर फॉर्म में कन्वर्ट किया जाता है. Now this powder of agarose, it is easily available from commercial companies and this agarose can be dissolved in a suitable buffer medium and then it is melted. जब ये मेल्ट हो जाए तो इसको अलो किया जाता है टू सोलिडिफाई एंड आफ्टर सोलिडिफिकेशन इसके ऊपर जो है वो डीएनए का सैंपल जो है वो लोड किया जा सकता है. Then there is another type of gel electrophoresis and it is polyacrylamide gel electrophoresis, and this gel it is mainly preferred for smaller DNA fragments. And even this is also used to isolate protein molecules. When we are using agarose to separate DNA fragments, so it is very important to adjust its concentration. For example, अगर हम छोटे DNA fragments को analyze करना चाह रहे हैं तो इसकी higher concentration use की जाती है like 2%, 3%, 4%. और अगर हम लार्जर डीएनए फ्रेगमेंट्स को एनालाइज करना चाह रहे हैं तो हम इसकी लो कंसंट्रेशन यूज़ करते हैं लाइक 0.7 to 1%. क्योंकि अगर हम लो कंसंट्रेशन यूज़ करेंगे तो इसका जो जेल का जो पोर्ट साइज है वो बड़ा होगा, दैट विल अलो larger molecules to pass through this gel. If we will use high concentration, the pore size will reduce and only smaller DNA fragments they can pass through this gel. Now, what are the different factors that can influence the mobility of the DNA through agarose gel electrophoresis? जैसे मैंने डिस्कस किया कि concentration of the gel it is very important, either higher concentration or lower concentration. Then the ionic strength of the buffer, जिस solution के अंदर हमने जेल को रखा है उसकी strength क्या है. अब ये जो buffer है इसकी composition different हो सकती है, like it may be TAE that is tris acetate EDTA or TBE that is tris borate EDTA. So ionic strength it is also very important to determine the mobility of the DNA. In addition to that, the voltage that is applied it is also important, and of course if ethidium bromide it is being used it is also important. Now what is ethidium bromide? Ethidium bromide actually एक स्टेन है, एक केमिकल है जो कि जेल को डाई करने के लिए यूज़ किया जाता है. So ethidium bromide can intercalate between the bases of double stranded DNA, and when it is illuminated to UV light then band of DNA they can be visible as orange color. So this is gel electrophoresis apparatus, and here this gel it is divided into two parts and it is placed in a reservoir that contain the buffer, and it is divided as I mentioned in two parts and where the DNA samples can be loaded. So here one can load the DNA and second part it is here. As DNA it is having predominantly negative charge, so from negative electrode the DNA will move towards positive electrode, and this will be the direction of the movement of DNA molecule under electric current. So here it is the visualization of DNA bands under ultraviolet light. If the gel is stained with ethidium bromide, then when it is exposed to UV light, then the bands of DNA they can be visible as orange color. So here it is the row of or the column of ladder, that is the marker that is run parallel with our DNA samples. So here these are the DNA samples that are visible as orange colored bands after exposure to UV light. Then in addition to exposure to ultraviolet light, now gel documentation systems, they are available by which you can visualize the DNA, and you can also save the pictures of your gel that you have run by gel electrophoresis. So again here these are different fragments within the marker. Marker means that we have the idea that what is the length of different fragments in that ladder that we run in one column at the right or left side. And then these are different bands that are separated on the basis of their length. For example, if the DNA fragment it is of large size, it will flow or travel slowly within the gel, and those DNA fragments that are of smaller size, they will flow more rapidly, so they will be at the base of the gel. So gel documentation system, it is an other addition where you can also record the results of your gel.